Welcome to this video in which we will see about the protein sorting in cell organelles. Introduction. Protein sorting, also known as protein targeting, is the process by which newly synthesized proteins are directed to specific locations within the cells. Eukaryotic cells contains many membrane-bound organelles such as nucleus, mitochondria, endoplasmic reticulum, Golgi complex, lysosomes and peroxisomes. Each of these compartments perform unique biochemical functions and therefore contains specific set of proteins and enzymes. Although proteins perform functions in different parts of the cells, most of them are synthesized by ribosomes located in the cytosol. Because of this, the cells requires a sophisticated system to transport proteins from the site of synthesis to the correct destination. Protein sorting is controlled by targeting signals embedded in the amino acid sequence of the protein molecules. These signals act as molecular addresses. Specialized receptors proteins recognize these signals and direct the proteins to the correct organelle through different transport mechanisms. Proper protein targeting is essential for cellular organization, metabolism, and communication between cellular components. When this process fails, proteins may accumulate in a wrong location, leading to cellular dysfunction and certain diseases. Now we will see the next point, that is intracellular compartments and protein distribution. Eukaryotic cells are divided into several compartments that allows different biochemical reactions to occur simultaneously without interfering with each other. Each organelle contains proteins that perform specific functions. Examples include the nucleus contains proteins involved in DNA replication, transcription, and DNA repair. Mitochondria contains enzymes which are responsible for energy production through oxidative phosphorylation. Lysosomes contain hydrolytic enzymes that digest macromolecules. Peroxisomes contain enzymes involved in oxidative metabolism and detoxification. The endoplasmic reticulum is involved in protein synthesis and lipid metabolism. Because proteins are required in many compartments, cell must sort them accurately to maintain proper organization and proper metabolic functions. In third point, we will see the mechanisms of protein transport. According to molecular biology of the cell, proteins are transported between cellular compartments by three major mechanisms. First one is gated transport. Gated transport occurs between the cytosol and the nucleus through structures called as nuclear pore complexes. These pores act as selective gates that regulate the movement of molecules across the nuclear envelope. Small molecules can diffuse freely through the nuclear pores, but large proteins require special signal sequences called as nuclear localization signals to enter the nucleus. The next mechanism of transport is transmembrane transport. In transmembrane transport, proteins cross membranes of organelles such as mitochondria, endoplasmic reticulum, chloroplast, peroxisomes. Protein channels known as translocators help protein move across the membranes. In many cases, protein must be unfolded to pass through these particular channels. The third mechanism is vesicular transport. Vesicular transport occurs between compartments of the endomembrane systems. In this mechanism, proteins are transported inside membrane-bound vesicles that bud from one organelle and fuse with the other one. This mechanism maintains the flow of proteins to the secretory pathway, which includes the endoplasmic reticulum, Golgi apparatus, lysosomes, endosomes, and plasma membrane. In next point, we will learn about signal sequences and targeting signals. Signal sequences are short stretches of amino acids that direct protein to their destinations. These sequences are recognized by receptor proteins that initiate transport. Signal sequences can occur at different positions within the protein molecule: N-terminal region, C-terminal region, and internal regions. In many cases, signal sequences are removed after the protein reaches to its destination. signal peptide. Protein destined for secretion or membrane insertion contains a hydrophobic signal peptides at the end terminal. This signal directs the protein to the rough endoplasmic reticulum. Nuclear localization signals. Proteins that function in the nucleus contain sequences rich in lysine and arginine, which are positively charged amino acids. These sequences allow proteins to pass through the nuclear pores. Mitochondrial targeting sequences. Mitochondrial proteins contain amphipathic alpha helices, which directs them to receptors on the mitochondrial surface. Peroxisomal target signals. Proteins destined for peroxisome contain specific sequences such as serine, lysine, leucine, PTS1 at the C-terminal end. These signals ensures that proteins are directed to the correct organelles. In next point, we will see details about the protein targeting to the endoplasmic reticulum. The rough endoplasmic reticulum is the entry point for proteins that follow the secretory pathway. These proteins include secreted proteins, membrane proteins, lysosomal proteins, proteins of the Golgi apparatus. Role of signal recognition particles. When a ribosome begins synthesizing a protein that contains a signal sequence, a molecule called signal recognition particle (SRP) binds to the signal peptide. SRP temporarily stops translation and guides the ribosome to the endoplasmic reticulum membrane. On the endoplasmic reticulum membrane, SRP interact with the SRP receptors. The ribosomes that attaches to the protein channel called as translocon SEC61 complex. Protein synthesis resumes and a growing polypeptide chain is threaded into the lumen of the endoplasmic reticulum. So, SRP particles, they take the ribosomes which is involved in synthesis of proteins on the membrane, and then the protein synthesis continues and the protein is transported in the lumen of the endoplasmic reticulum. Protein folding and quality control. Inside the endoplasmic reticulum, protein undergoes several modifications, folding with the help of molecular chaperones. Chaperones are the proteins which are help the proteins for folding and unfolding, formation of disulfide bonds, addition of carbohydrate chains through N-linked glycosylation. Endoplasmic reticulum also contains quality control system that ensures only properly folded proteins leave the organelles. Misfolded proteins are transported back to the cytosol and degraded by proteasomes. Proteasome is the machinery present in eukaryotic cells which is involved in degradation of proteins. Ubiquitin is a tag when it is attached to the protein in a form of polyubiquitination. The proteins are destined for the degradation by proteasomes. The next point is protein processing in the Golgi apparatus. After leaving the endoplasmic reticulum, proteins are transported to the Golgi apparatus. The Golgi consists of flattened membrane sac-like structures known as cisternae, arranged into three main regions: cis Golgi, medial Golgi, and trans Golgi network. A protein moves through the Golgi stacks. They undergo several modifications including trimming of carbohydrate chains, addition of sugar residues, phosphorylation and sulfation. The Golgi apparatus also act as a major sorting center of the cell, where the proteins are sorted and transported to destined organelles. Vesicular transport in detail. Vesicular transport is one of the most important mechanisms of protein sorting. It allows protein to move between compartments without mixing the content of different organelles. Transport vesicles are small membrane-bound sacs that carry proteins from one organelle to another organelle. Steps in vesicular transport. Cargo selection. Cargo is something that need to be transported. So first point is cargo selection. Specific proteins are selected for transport using cargo receptors which are present in the membrane-bound vesicles. These receptors recognize sorting signals on the proteins and concentrate them in the budding vesicles. Vesicle budding. Vesicles from these coat proteins assemble on the cytosolic side of the membrane. These coat proteins bend the membrane and help them to form the vesicles. Three major coat proteins which are involved are COP2 vesicles. COP2 vesicles transport proteins from endoplasmic reticulum to Golgi apparatus. Important components include SAR1 GTPase, SEC23, SEC24 complex, SEC13 and SEC31 coat proteins. SAR1 initiates vesicle formation by inserting into the endoplasmic membrane and recruiting coat proteins. COP1 vesicles. COP1 vesicles transport proteins from the Golgi complex back to the endoplasmic reticulum. This is known as retrograde transport. This transport is important for recycling of endoplasmic reticulum proteins that accidentally escape to Golgi complex. Lathrin coated vesicles. Lathrin vesicles transport proteins from Golgi to lysosomes and endosomes. Lathrin forms cage-like structures around vesicles that helps shape the budding vesicle. Adaptin proteins connect lathrin to cargo receptors. The next point is vesicle targeting and fusion. After vesicles bud from the donor membranes, they must find the correct target membranes. RAB-GTPase, RAB protein act as a molecular marker that help guide vesicles to the correct destination. Each organelle has specific RAB proteins that ensures vesicles fuse only with the correct membranes and correct organelles. Tethering proteins. Tethering proteins capture vesicles when they arrive near the target membranes. SNARE proteins. SNARE proteins are responsible for membrane fusion. Two types of SNARE proteins participates, vSNARE on vesicle membranes and tSNARE on target membranes. When these proteins interact, the vesicle membranes and target membranes are pulled together and they fuse with each other. This fusion releases the cargo protein into the target organelles. Now we will see details about the protein sorting to mitochondria. Most mitochondrial proteins are synthesized in the cytosol and imported into the mitochondria after translation. These proteins contain mitochondrial targeting sequence. Transport occurs through two main protein complexes, TOM complex, the translocase of the outer membrane, and that's why the short form is TOM. O stands for outer membrane. Allows protein to cross the outer mitochondrial membrane. TIM complex, the translocase of the inner membrane, transports proteins across the inner membrane. Energy for this process is supplied by ATP and membrane potential across the inner membrane. Once inside the mitochondrial matrix, the targeting sequence is removed and the proteins fold into the functional forms. Now we will see details about the nuclear protein transport. In case of nuclear protein transport, proteins required in the nucleus are transported through nuclear pore complexes. These proteins contain nuclear localization signals. Transport receptors called importins recognize these signals and binds to the protein. The protein importin complex moves through the nuclear pore and releases the protein inside the nucleus. The process is regulated by the small GTP binding protein called as RAM, which controls the direction of nuclear transport. Protein sorting to peroxisomes. Peroxisome contains enzymes involved in oxidative metabolism and detoxification. Proteins are targeted to peroxisomes by peroxisomal targeting sequences or signals, PTS. Two types exist, PTS1, serine, lysine, leucine sequence at the C-terminal end, and PTS2, N-terminal targeting sequence. These signals are recognized by receptor proteins such as PEX5. Unlike mitochondrial and ER, peroxisomes can import fully folded proteins. Biological importance of protein sorting. Protein sorting is essential for maintaining cellular organization and metabolic efficiency. Correct protein targeting ensures that enzymes interact with proper substrates, metabolic pathways occurs in the correct compartment, signaling pathways function properly, and proteins are secreted when they are needed outside the cell. Errors in protein sorting can lead to several diseases including eye cell disease, Zellweger syndrome, and neurodegenerative diseases related to protein misfolding. Coming to the conclusion of this particular topic, protein sorting is a complex and highly coordinated cellular process that ensures proteins reach the correct intracellular destination. Through the combined action of signal sequences, receptor proteins, translocation channels, and vesicular transport systems, proteins are accurately delivered to organelles such as ER, Golgi apparatus, mitochondria, nucleus, peroxisome. These mechanisms maintain cellular organization, support metabolic functions, and ensure that proper cell physiology is maintained. Thank you.